Hi students, this is Mr. Eckert. We're going to go through target C14, which for many of you will be pretty much a review target. You can see here the target says that you should be able to create an accurate model of the DNA molecule. And we've talked about models already, and you probably remember that a model can be something like a plastic model, or it can be a drawing, or a written description can be a model, or even a mathematical model. So it says before you can create an accurate model of DNA, you have to know a little bit about the structure and the function of the molecule. So what is DNA? You might remember from The Simpsons, they had an answer.
Just ask this scientician. Just ask this scientician.
So obviously that guy didn't know very much at all, but you probably do know that DNA is the molecule of heredity. It contains all the information that makes you you. It has all the instructions for making every single protein that all of your cells will ever make, and that's about 2 million different kinds of proteins. So what does it look like? Here's a computer image of a DNA molecule, which you probably recognize as, even in this, you can kind of see that double helix shape. But it consists of long strands of submolecules or subunits called nucleotides that are bonded together. And you can see here a repeating pattern in this artist's rendition. So you can see PSB kind of repeats. There's 3 of these units. So those are the nucleotides. Every nucleotide has 3 parts then. A sugar, and DNA's sugar is called deoxyribose. In fact, that is the D in DNA. It stands for deoxyribonucleic acid. So every nucleotide has a sugar, one phosphate. And there is a difference between a phosphate and a phosphorus. A phosphorus atom bonded together with 4 oxygen atoms make up a phosphate. And then there's one nitrogen base, and you probably remember there's 4 different nitrogen bases. Their names are adenine, thymine, cytosine, and guanine. Usually those are abbreviated to the first letters A, T, C, and G. So you can see in this diagram here's a G, a guanine attached to a sugar, and you'll want to remember that the nitrogen base is always attached to only the sugar. You can see that that's true in every diagram that you'll ever see of DNA. So we've got a guanine attached to a sugar, attached to a phosphate, so this is one nucleotide right here. So DNA consists of just a long chain of those, much like a train consists of a chain of cars. The cars then would be the nucleotides. So DNA molecule, I'm sure you've heard before, is called a double helix. It looks like a ladder that's been twisted into a spiral, and the sides of those ladders are made up of alternating sugar and phosphate molecules. It just goes sugar, phosphate, sugar, phosphate, sugar, phosphate all the way down the sides. And then the rungs of the ladder in between are made up of those base pairs of the nitrogen bases, A, T, C, and G. And again, those attach to the sugar molecules on the side. The base pairs, or the nitrogen bases, they bond together with what's called a hydrogen bond that holds them together, and they follow what's called a base pair rule, and that is that A always bonds with T, and the opposite is true. T always bonds with A. C always bonds with G, and G always bonds with C. And so you might ask the question, well, why is that? And it has to do with the geometry and the chemistry of those molecules. The adenine and the thymine, they both can form only 2 hydrogen bonds between the 2 of them, while the cytosine and the guanine, they form 3 hydrogen bonds. So one tip that'll help you to remember that, this helps me, is when you write a capital A, of course you're using all straight lines. When you write a capital T, you're using all straight lines. When you write a C or a G, you're using a curvy line. So A bonds with T, T bonds with A, C bonds with G, and G bonds with C. Now we say that those 2 strands of DNA are complementary to each other, and it's actually, of course, the nitrogen bases that are complementary. And we don't mean complementary in the way that you would give somebody a compliment, like, oh, your hair looks nice, or that's a nice outfit you're wearing, but complementary in terms of kind of like 2 puzzle pieces that fit together. Or you may remember when we studied enzymes and substrates, the active site on the enzyme was complementary to the substrate. The shapes, they fit together like puzzle pieces. So then, if you know one side of a DNA molecule, like this right here, the sequence of nitrogen bases, then you should be able to predict what the other side would be. So take a look at this. I believe you have this in your notes. And just take about 10 seconds to go ahead and add the other complementary side that would be opposite to this. And you may want to pause the video at this point and restart it when you're done writing that down in your notes. So if you've done that, of course this is what your answer should be, but if you look at this, you should see one mistake in it. And can you spot it? And it of course is right here. So A bonds with T, T bonds with A, C bonds with G, so this should be a G here, and the rest of it is correct. So, and of course you probably have the correct answer in your notes. Now last, since A always bonds with T, A and G are known as purines. You can see the structure of those molecules consists of two rings. There's a ring here and a ring here. And in adenine, a ring here and a ring here. So adenine and guanine are known as purines. Those are two-ring molecules, whereas thymine and cytosine are known as pyrimidines. So since A always bonds with T, and you can see the pyrimidines only have one ring in the T and the cytosine. So since A always bonds with T, a purine always bonds with a pyrimidine, and that's true when G bonds with C as well. So that does it for this target. You probably have already done the DNA coloring worksheet. If you have not yet done so, you'll find it in the assignment folder for this set of targets. And if you have any questions, make sure you ask.